Alright, so we're going to pick up right where we left off last time. We had just looked at a little bit about Mendels Experiment with peas and things like that, and we got some useful terminology like alleles, dominant, and recessive out of the way. And what we're going to do is we're going to look a little bit more closely at the results of his experiments today to introduce some new terms like Punnett squares, homozygous, heterozygous, things like that. So again, this is just, given the cell processes unit assessment, is just very, very basic, probably a review for most of you, but something I wanted to put out there so that, especially if it's something from the diagnostic assessment or the diagnostic for heredity, that if you need to work on, that there's some resources out there for you. So, continuing with those words from last time, okay? Mendel's experiment we could reframe like this. So we had talked about how the P, the F1 and the F2 generation last time, okay? The two flowers he started with in the P generation that were true breeding purple and true breeding white flowers, we can call those their phenotypes. Their physical manifestations, what they looked like. All right? We could say that their genotype is, and the true breeding purple flower would have two dominant P's because it's true breeding, meaning always producing offspring that look like itself. That's why it has two dominant P's. And the true breeding white flower has two recessive P's. So that's another way to say true breeding is a organism that has two of the same alleles, whatever that is, two dominant or two recessive. So these are both true breeding because the purple one has two dominant alleles and the white one has two recessive alleles. So then when he crossed these, okay, when he crossed them, we get one allele from each parent. So this purple flower donated a dominant allele. This recessive flower donated a recessive allele. That's why all of the offspring looked the same. They had one dominant allele and one recessive allele. And since the dominant allele is always expressed, that's why 100% of the offspring looked purple. But then when they self-pollinated these flowers and got 75-25, what would be the genotypes? We know the phenotypes, purple and white. What would be the genotypes to come up with this 3-to-1 ratio? And so we can actually determine that, the likelihoods, the probabilities of those using a Punnett Square. A Punnett Square. And this is a way to calculate the probability of having offspring with different genotypes and different phenotypes if you know the genotypes of the parents. So it kind of looks like this. So if we were going to cross these two purple flowers that each had a dominant allele and a recessive allele, for one side of the square, usually on this side, one side is going to be for each parent. And there will be a column for each allele that they could potentially contribute to their offspring. So if I take this male flower and put it on the top, it could contribute a dominant P. I'll put that over one column. And it could contribute a recessive P. I'll put that over a second column. And then for the second parent, we'll put along the side, and there'll be a row for each potential allele that they could contribute. So there would be a row for the dominant P and a row for the recessive P. And then to figure out the potential genotypes of the offspring, we just match up alleles from the same column and the same row. So this is in the first column, this is in the first row, this is one possibility. The second column matched with the first row is another possibility, and so on and so forth. This square represents the probability or the proportion of offspring that would have each genotype and each phenotype. So, we would expect 75% of them to be purple and 25% of them to be white as far as the phenotype in this column. We would expect 25% of them to be true breeding purple flowers, so 2 dominant P's, 25% to be true breeding white flowers, so 2 recessive P's, and 2 of them, or 50%, to be purple flowers that had 1 allele of both. So notice that the genotype ratios and the phenotype ratios can be different. Genotype has to do with what specific combination of alleles we have. Phenotype is just, in this case, what color. All right. So, extending the idea a little bit farther. So we said that true breeding means having 2 of the same allele, 2 dominant or 2 recessive. The genetics word we use for that is homozygous. Homozygous, meaning that the same alleles, 2 dominant or 2 recessive. So we call this genotype with 2 dominant alleles homozygous dominant. Homozygous dominant. And 2 alleles that are recessive, homozygous recessive. If there is a genotype that has 1 of each allele, 1 dominant and 1 recessive, we call that heterozygous, for different alleles. Okay, so in our pea plant here, the offspring, 25% were homozygous dominant, 25% were homozygous recessive, and 50% were heterozygous. That's a better, more accurate way to describe them. So 2 organisms can have the same phenotype but different genotypes. For example, in the purple flowers, there could be a homozygous dominant flower that is purple, or there could be a heterozygous dominant, or not dominant, heterozygous flower that is purple. And both of them are purple because both of them have at least 1 dominant allele, and anything that has at least 1 dominant allele will have a dominant phenotype. There is no way in Mendelian inheritance. We'll talk about non-Mendelian inheritance after the break, but in Mendelian inheritance, there's no way to visually look at and tell the difference between these 2. In genetics, there is something that's extremely important in especially doing genetic experiments or genetic experiments, especially when you're crossing or breeding 2 different organisms together, and it's called a test cross. And it is when you breed the dominant phenotype with an unknown genotype. When you breed a dominant phenotype, so an organism that you know has a dominant trait, but you don't know whether it is homozygous or heterozygous, and you breed that organism with another that you know is homozygous recessive. So this is called a test cross. So in your book and my videos going forward, when it says that you're completing a test cross, that means you're breeding something that you know it is a dominant phenotype, but you don't know its genotype, and you're crossing it with a known homozygous recessive individual. And by doing this, you are able to determine the identity of the unknown allele. You'll be able to figure out whether this flower is homozygous dominant or homozygous recessive. So let's see how that would work. So here is a test cross, and let's say in both cases we don't know what the purple flower is. It could be homozygous dominant or heterozygous. If we cross it with a homozygous recessive flower and the purple flower is homozygous dominant, well then we're going to get all purple flowers. All offspring have the dominant phenotype. If it was homozygous, or heterozygous, we would get 50-50. Half of our offspring would have the dominant phenotype, half would have the recessive. So by looking at the ratios of the phenotypes of the offspring, we can determine what the genotype of that parent was. This is why a test cross for kind of simple Mendelian type genetic experiments is extremely useful. Right. I think that's as far as I want to go with today, is just talking about simple Punnett squares and talking about test crosses and homozygous and heterozygous. We'll get into more with heredity when we come back next time.